Okay, now we will see about this acquired immunity. It is also known as the adaptive immunity. Acquired immunity is the ability of the body to defend itself against invading foreign agents through specific defense responses mediated by diverse T lymphocytes and B lymphocytes. So acquired immunity, it shows specificity for particular foreign molecules. Recognition of the animal's own molecules from non-self and memory for previously encountered pathogens. So that subsequent encounter causes stronger and more rapid response, which is also called immunological memory. We'll come to this in detail. So in the animal kingdom, acquired immunity is found only in the vertebrates. So cells that have developed the ability to carry out acquired immune responses, if they are activated against foreign agents, are the T lymphocytes and the B lymphocytes. So here we have to remember the definition of the acquired immunity, which is the ability of the body to defend itself against invading foreign agents through specific defense responses mediated how? By T lymphocytes and B lymphocytes. So in acquired immunity, T lymphocytes and B lymphocytes are playing a very important part in the defense. So it is a specific defense response, not like innate immunity, which is a non-specific defense. Now acquired immunity, it also shows specificity for the foreign molecules, recognition of self from non-self and memory, and found only in the vertebrates, and the cells that develop the ability to carry out the responses are the T lymphocytes and the B lymphocytes. So this is what you have to know in this particular slide. Okay, now we will see about this acquired immunity. It is also known as the adaptive immunity. Acquired immunity is the ability of the body to defend itself against invading foreign agents through specific defense responses mediated by diverse T lymphocytes and B lymphocytes. So acquired immunity, it shows specificity for particular foreign molecules. Recognition of the animal's own molecules from non-self and memory for previously encountered pathogens. So that subsequent encounter causes stronger and more rapid response, which is also called immunological memory. We'll come to this in detail. So in the animal kingdom, acquired immunity is found only in the vertebrates. So cells that have developed the ability to carry out acquired immune responses, if they are activated against foreign agents, are the T lymphocytes and the B lymphocytes. So here we have to remember the definition of the acquired immunity, which is the ability of the body to defend itself against invading foreign agents through specific defense responses mediated how? By T lymphocytes and B lymphocytes. So in acquired immunity, T lymphocytes and B lymphocytes are playing a very important part in the defense. So it is a specific defense response, not like innate immunity, which is a non-specific defense. Now acquired immunity, it also shows specificity for the foreign molecules, recognition of self from non-self and memory, and found only in the vertebrates, and the cells that develop the ability to carry out the responses are the T lymphocytes and the B lymphocytes. So this is what you have to know in this particular slide. Now in man, both types of lymphocytes are originated from the stem cells in the bone marrow. So there are certain cells which in the bone marrow which we call stem cells. So these T lymphocytes and B lymphocytes, they have originated in the stem cells, and some of these, they have migrated to the thymus for maturation and are called T lymphocytes. So they are called T lymphocytes because they have migrated to the thymus for maturation. and those lymphocytes that remain in the bone marrow to complete the development, we call them the B lymphocytes. Okay, so T lymphocytes and B lymphocytes both have been produced in the stem cells of the bone marrow, but because the T lymphocytes, we call them T lymphocytes because they are migrated to the thymus for maturation, and B lymphocytes because they remain in the bone marrow for the completion of the development. Now, before leaving these lymphocytes to the secondary lymphatic tissues, for example, the lymph nodes and the spleen and all, their plasma membrane, they acquire diverse specific protein receptors. We will come to that and show you some pictures about that. So these specific protein receptors, they are called antigen receptors. So antigen receptors have the ability to recognize the specific foreign agents or foreign invasions. Now, it has been found that there are over 100,000 antigen receptors on the surface of a single B lymphocyte or a T lymphocyte. Okay, so in this slide you have to know about the T lymphocytes and the B lymphocytes, why we call them T lymphocytes and B lymphocytes, and the antigen receptors, the acquisition of antigen receptors on these lymphocytes. Okay. Now, what is an antigen? What is an antigen? An antigen is a substance that has the ability to stimulate an immune response through T lymphocytes and B lymphocytes and to react with specific cells or antibodies that resulted from the stimulated immune response. So, only if there is an antigen, an immune response can take place. Okay, so it is a substance which has the ability to stimulate an immune response through the T lymphocytes and the B lymphocytes. Okay, and after that it has the ability to react with the antibodies that have resulted from this stimulated immune response. So the antigen is the one that has the ability to stimulate an immune response through these lymphocytes. Now here, what are the antigen? If you say, examples of antigen: viral proteins, bacterial toxins, chemical components of bacterial structure like flagella, cell walls, they are all antigenic. We say that they are antigenic. Structural components of incompatible blood cells. Okay, during transfusion, you find that the plasma membrane, transplanted tissues can be antigenic. So, structural components of the incompatible blood cells. So, when they are given a wrong transfusion, okay. So, these are all these act as antigens. These are examples of antigens. Now, antigens are usually large foreign molecules such as proteins and polysaccharides. What are they are for large foreign molecules. So the chemical composition, they can be proteins and polysaccharides. So not the entire antigen, but certain parts of a large antigen molecule act as triggers for the acquired immune response. So no need of the entire antigen, but even small parts of it can trigger an immune response. Okay. So in this slide you have to know what an antigen is. It has the substance which has the ability to stimulate an immune response through the T lymphocytes and B lymphocytes and react with these antibodies, specific the cells which have resulted from this immune response, the different types of antigens, okay, and the chemical composition of these antigens, okay, and only a part of the antigen can stimulate an immune response, no need of the entire antigen. So then we can go to the next slide. Yeah. So you that is clear. So in this slide you can see about the antigen, the epitope, which binds to a T lymphocyte, the antigen receptor of a T lymphocyte or a B lymphocyte. So this single antigen can have several epitopes and each can bind with the specific antigen receptor. So the antigen receptor is very specific to that particular type of a epitope of a single T or B lymphocyte. Okay. Then we can go. Here you can see the structure of the T lymphocytes and the B lymphocytes. So you have a mature B cell or a B lymphocyte, and you have a mature T cell or a T lymphocyte. You can identify them by the type of the antigen receptors which are found on the surface of the plasma membrane. Now in the mature B cell you find the Y-shaped antigen receptor, and the T lymphocyte, the structure is different. Okay. Here you find the mature B cell and the mature T cell with the antigen receptors. And you find the antigen receptor of a B cell binding to the epitope of that particular antigen. Cell-mediated and humoral response. So in acquired immunity, two types of immune responses are there. They are mediated by T lymphocytes and B lymphocytes. So you get the two responses are the cell-mediated immune response and the humoral response. Now humoral response is also called antibody-mediated immune response. Okay, so antibodies are involved here. Cell-mediated immune response, it is mediated by the T lymphocytes, and here in this response, these cells, the T lymphocyte, they directly kill the infected cells. Whereas in the humoral immune response, antibodies are secreted into the blood and the lymph, and they mark the antigen for deactivation. Okay. So both these immune responses are triggered by antigens. Okay. So any pathogen, they can provoke both type of the immune responses, cell-mediated immune response and humoral immune response. Cell-mediated immune response is a type of acquired immunity in which specifically sensitized T lymphocytes attached to the antigen undergo proliferation and differentiate into cytotoxic T cells. So here the T lymphocytes have to be activated, sensitized or activated. So what happens is that these T lymphocytes, they attach to the antigen. When they attach to the antigen only, they get sensitized. They proliferate. That means they undergo multiple divisions to form a clone of cells, which are these cells are exactly like each other. A group of cells which are exactly like each other. And eventually differentiate into cytotoxic T cells. So these activated T lymphocytes attach to the antigen, undergo proliferation and differentiate into cytotoxic T cells. This is the process that happens once these T cells are activated. And what happens is these cytotoxic T cells, they directly kill the cells which are infected or kill the cells with the invading antigen. Now in addition, what happens, memory T cells also are formed. Some cells of these clone, they form the cytotoxic T cells which take action immediately and kill off these infected cells. Then some of them form the memory cells also, which are found in the body, and they can cause a stronger and more rapid response at subsequent encounter of the same antigen. That means that the same antigen enters the body later on, after a few years or many years. These memory cells, they can cause a stronger and rapid response when they meet the same antigen even after that. Okay, so this is what we call the immunological memory. So this is effective against the infected cells, cells infected by fungi, parasites, virus. Okay, all these present within the host cells, some cancer cells, foreign transplanted cells. Now, the main thing here is that cell-mediated immunity, it involves cells attacking cells. Cells attacking cells. So, these cytotoxic T cells, they are killer cells. They directly kill the cells which are infected, and some of these cells in the clone, they form memory cells which can um, form produce a stronger response at the subsequent encounter of the same antigen. You have to write it like that. Okay. Now here you have these T lymphocytes. You see the T cell antigen receptor and the structure of the T cell antigen receptor. This is not necessary for you, but you can just have an idea about these antigen receptors. Is made up of these alpha and beta chains, and this antigen binding site. Look at the shape of the antigen binding site. Now here this host cell has been infected by a pathogen, and some fragments of these pathogens are found in the host cell. Now what happens is that one MHC molecule, MHC means major histocompatibility complex, which is in green color, that binds to this fragment, antigen fragment, and displays it on the surface of the host cell. Okay, displays it on the surface of the host cell. Therefore, when it is displayed like that, these T cells, they come and they can attach. Okay, they can attach to this the antigen receptor can bind to the MHC molecule which carrying the antigen fragment. Okay, so this is how the T cell is able to identify the antigen. Okay, so this MHC molecule and the antigen fragment, with the antigen fragment, it is able to bind to the T cell. Okay, so here you have a view of this MHC molecule with the antigen fragment. So this shows how this antigen fragment is cradled by the MHC molecule. Humoral immune response is a type of acquired immunity in which the specifically sensitized B lymphocytes attached to a particular antigen undergo proliferation and eventually differentiate into plasma cells that secrete the circulating antibodies that can neutralize and inactivate the specific toxins and pathogens in the blood and lymph. Now, humoral immune response or antibody mediated immune response, which it is called, okay, it is specifically in which specifically sensitized B lymphocytes. Now these B lymphocytes, they attach to the antigen, undergo proliferation and differentiate into plasma cells. Okay, plasma cells are the effector form of this B cells. They take effect immediately. So they are found in the, so what happens, these plasma cells, they secrete the antibodies. Okay, those Y-shaped antibodies, antigen receptors, they are the antibodies. So the plasma cells secrete the antibodies into the blood and the lymph tissue fluids, and they do not kill the cells, but they can neutralize and inactivate the toxins and pathogens in the blood and the lymph. Now, in addition to this, I told you some memory cells also are formed, which can cause stronger and more rapid response at subsequent encounter of the same antigen. Same thing we found in the cell-mediated immune response also. Memory cells also are there, found there. So, we call this immunological memory. Now, immune response works mainly against antigens present in the body fluids and extracellular pathogens, mainly the bacteria, which multiply in the body fluids. Okay, so this response is against the antigens and the extracellular pathogens found in the body fluids. Here you have a picture about this humoral immune response, where you have the antigens bind to the antigen receptors of only one of the three B cells. So you have three types of B cells. You see the antigen receptors are different in the shape. So this antigen receptor, so the antigen binds, only this particular antigen receptor can bind to the antigen because of the shape of it. Okay, it is very specific. The selected B cell, so this one, once it binds, it gets activated and it starts to proliferate, forming a clone. So a clone of identical cells, similar cell, not similar, identical cells, bearing receptors for the antigen. So it proliferates into a clone of cells, and some of the daughter cells develop into long-lived memory cells, which can bring about rapid response upon subsequent exposure. That means after some years, they can, if they are confronted with the same antigen, they can become activated and, they can destroy the antigen. And other daughter cells develop into short-lived. So these are long-term. They have long-lived memory cells, which we call immunological memory here. And other daughter cells develop into short-lived plasma cells, which secrete the antibodies specific for the antigen. Okay. So these antibodies can neutralize or deactivate that particular pathogen which is present in the blood and the body fluids. Okay. So you have immunological memory also, and here you have these plasma cells which take effect immediately. These are very short-lived cells uh, which secrete the antibodies. Okay. Now, what are these antibodies? Antibodies are proteins secreted by plasma cells. Okay, the plasma cells are differentiated B lymphocytes. So the antibodies are proteins which are secreted by the plasma cells in response to specific antigens. We've seen that in that slide. So antibodies, they are proteins secreted by the plasma cells in response to the antigen. Just simply they are not secreted, in response to the specific antigen. Now the antibody binds with the antigen to neutralize, inhibit or destroy it. Okay, the antibody binds with that antigen to neutralize, inhibit or destroy it. Okay, so they can neutralize, inactivate the toxins and the pathogens in the body fluids, specific toxins in the body fluids. The antibodies do not directly kill the pathogen. Okay, not that. They do not directly kill the pathogen, but can interfere with the activity of the pathogen or mark the pathogen for inactivation and destruction. That you have to know. They do not kill the pathogen, but they interfere with the activity of the pathogen and mark the pathogen for inactivation and destruction. Antibody antigen complexes can activate. Now how do these antibodies destroy the pathogen? So these antibody antigen complexes, they can activate another system called the complement system. You would have seen that in the innate immunity. Okay, the complement system. Okay, and phagocytosis to destroy the pathogen. So they activate these to destroy the pathogen. So antibodies are also called immunoglobulins. Note that antibodies are also called immunoglobulins. And the immunoglobulin has the same Y-shaped structure as B lymphocyte antigen receptors. So they have the same Y-shaped structure as B lymphocyte antigen receptors, but they are secreted rather than membrane bound. So these Y-shaped structures, these antibodies, they are the B lymphocyte antigen receptors. Okay, they are secreted rather than membrane bound. Okay. So here you have to know what an antibody is. They are a protein secreted by the plasma cells. They differentiate B lymphocytes in response to specific antigens. Antibody binds the antigen and neutralize or inhibit or destroy it. They do not kill the pathogen but interfere with the active pathogen and mark the pathogen for inactivation and destruction, and they activate the complement system and phagocytosis, destroy the pathogen. So they are also called the immunoglobulins. They are the Y-shaped structures. Okay. Here you can see the antibodies which are secreted once the B lymphocyte antigen receptor binds to the epitope. Proliferation takes place. The plasma cells, they are called the effector cells, which a clone of them are produced. They proliferate and they secrete the antibodies into the blood and the lymph, which can go and bind to the epitopes of the antigen and pathogen and destroy the pathogen. Okay, they can mark the antigen for inactivation. They can mark the antigen for inactivation and destroy it, activating the complement system of phagocytosis. Okay, so you find antigen A, B, C. They have different structures. Okay, so they are very specific, in which they bind only to that particular type of the epitope. Okay, now we will see about the role of T lymphocytes and B lymphocytes in acquired immunity. Now these are the 4 steps you have to remember. The role of the T lymphocytes and B lymphocytes in acquired immunity. How do they function? So now the first step is the recognition of the antigen, binding of the antigen and sensitization. Okay, recognition, binding and sensitization. So these cells have to recognize the antigen first and bind to it and become activated. So when these lymphocytes, they bind to the antigen only, they become activated. So these cells, T lymphocytes and B lymphocytes, they can bind to the same antigen, but the way they recognize the antigen is different. So that we have to go and see into this. Okay. So first step, recognition, binding and sensitization. Okay. And second step is the proliferation and differentiation into effector cells. Proliferation, as I told you, the multiple division of the cells to form identical, a clone of identical cells. Okay, and they have to differentiate into effector cells. Effector cells are cells which take effect immediately to destroy the pathogen. Effector cells take effect immediately to destroy the pathogen. Okay, and then the elimination of the invaders, how they get rid of the pathogen. The technique they involve in getting rid of the pathogen, and the immunological memory which is provided by these T and B lymphocytes. So these 4 ways in which the role of the T lymphocytes and B lymphocytes are going to be, we are going to see it one by one. Okay, so the recognition, the first step is the recognition of the antigen, binding to the antigen and sensitization or activation. Now, recognition of the antigen occurs through a successful match between the epitope of the antigen. Okay, you know what an epitope is, a small accessible portion of the antigen which binds to the antigen receptor. Okay, so a match, so it should match the epitope of the antigen and the antigen receptor on a small number of B lymphocytes and T lymphocytes. Okay, so the antigen should match the antigen receptor. So as specific antigen receptors produced by single T cell or B cell can be identical, they can bind to the same epitope. Okay, so they can target the same epitope or the same antigen. Okay, but both the T and B cells respond to the pathogen, can respond to any pathogen that produces molecules containing the same epitope. Okay, so these T and B cells, they can respond to any pathogen which contains the molecules with the same epitope. But the way the B cells and T cells encounter the antigen is different. Okay, the way the B cells and T cells encounter antigens is different. So in this slide, you remember the recognition of the antigen, binding of the antigen, and sensitization. Okay, so the recognition occurs through the match between the epitope and the antigen on a small number of antigen receptor on the B lymphocytes and T lymphocytes. Okay, so now this is the main thing in this slide, that the B cells and the T cells encounter the antigen in different ways, although they can bind to the same epitope. Okay. Now this is the recognition of antigen by the T cells. Now here we have already seen this, where the host cell engulfs the pathogen and the fragment of the antigen is displayed by the MHC molecule. Okay, and here you find when this fragment is displayed, this T cell or the T lymphocyte antigen receptor will recognize and bind to this. So this is the way the T cell is able to recognize the antigen. Okay, so this antigen has to be presented to the T cell on the surface by this MHC molecule, only then the T cell will recognize and bind to it. Now I told you the B cell will recognize in a different way. Here there are three different types, say A, B, C. Okay, now this antigen can bind, so the epitope of this antigen can bind only to this particular B cell. Okay, so the specificity of the B cell is different. Okay, so the antigen specificity is different here. So this, once the antigen binds to the antigen receptor, the cells, the B cells, they proliferate to form a clone. Some of them form effector cells. The effector form of the B cells are the plasma cells, which secrete the antibodies, which can go and bind to the antigen, or some of the cells in the clone can produce memory cells, which are they are long-lived cells, long-term cells, or long-lived cells, which can produce a stronger immune response at a later time if the same antigen is confronted. Okay. Now here, recognition of the antigen, binding to the antigen, and sensitization. So T lymphocytes only recognize fragments of antigen protein that are presented to cells by special cells called antigen presenting cells. We seen this in the previous slide. Now these antigen presenting cells, they can be macrophages, okay, they can be dendritic cells and B cells. Now B lymphocytes can recognize and bind to antigens present in the blood, lymph and interstitial fluid. That also I told you earlier. Okay, so T lymphocytes, they have to have these antigen presenting cells by which an MHC molecule is able to display the antigen fragment. Okay, so they can recognize the fragments of the antigens which are presented by certain cells. But B lymphocytes, they can bind to the antigens in the plasma and blood and interstitial fluid. So the binding of an antigen to the antigen receptor results in sensitization. So once the antigen Once the B cell or T cell binds to the antigen only, they become activated. Okay, sensitization, activation of this specific T lymphocyte or B lymphocyte, and this activation initiates the cell-mediated antibody immune response. Okay, cell-mediated antibody or cell-mediated and antibody mediated immune response. Okay. Now here you have the dendritic cell. Dendritic cell, you have see these dendrite-like structures here. Okay. Now this is an antigen. This is also an antigen-presenting cell. This is engulfed in an antigen and displays it on the surface by an MHC molecule. And you have an accessory protein also called CD protein. Okay, the antigen receptor of this T cell, helper T cell. We'll speak about these helper T cells a little bit later. And then the antigen receptor, it binds to this complex, this antigen fragment. Okay. So once it binds only, this T cell becomes, this helper T cell becomes activated, and some cytokines are produced by this dendritic cell, which activates the helper T cell, and these helper T cell also produces some cytokines, which again in turn activates the helper cell, and again these cytokines, they activate the cytotoxic T cell, which brings about cell-mediated immunity, and they can also activate, these cytokines can also activate the B cell, which brings about the humoral immunity. Right? Now, proliferation and differentiation into effector cells. We've seen how they recognize the cell, how they recognize the antigen, and they bind to it, and they activate it. Now what happens after that? So once activated, the T lymphocyte and B lymphocyte undergoes multiple divisions, which we call proliferation, and result in a clone, a population of cells that are identical to the original lymphocyte. And some cells of the clone become effector cells, which are short-lived and take effect immediately against the antigen to provide primary immune cells. So I told you about these effector cells. So after activation, they proliferate to form, that means they undergo multiple divisions to form a clone of cells which are identical to the original lymphocyte, and some of the cells become effector cells which take action immediately against the pathogen to destroy it. They are short-lived cells, and these effector cells only provide what is called primary immune response. They provide the primary immune response. Okay. Now here, the effector T cells and B cells and memory cells. I spoke to you about the effector cells in the previous slide. Now, the effector form of the B cells are the plasma cells. They take effect immediately. So the plasma cells, a clone of these plasma cells are formed, which are all similar to the original cell, B cell. And these plasma cells, they secrete the antibodies into the blood and the lymph and interstitial fluid, which go and bind to the antigen and mark it for inactivation or destruction. Right? Some of these cells, effector cells, in the clone, some of these cells, they become memory cells. Some of these cells become memory cells. See, some of them differentiate into plasma cells, which are the effector cells. The effector form of the B cells are the plasma cells, which take effect immediately. Some of them become the memory cells, and which are the long-term, long-lived cells. And if the same antigen is met some years later, they produce a more stronger and rapid response. So they become effector cells if they confront the same antigen or they meet the same antigen again. Okay. So the effector cells, they take effect immediately, while memory cells, they are long-lived cells which can differentiate into effector cells if they come across the same antigen again later in life. Now, the effector T cells, the effector form of the T cells, these are the cells that short-lived cells that take effect immediately. Effector form of the T cells are the helper T cells and the cytotoxic T cells. Effector form of the T cells are the helper T cells and the cytotoxic T cells. Okay, so these take action immediately and kill the infected cells, while some of these cells in the clone, they form memory cells which are identical to the original cell, and if the same antigen is met later or confronted later after some time, they again differentiate into the effector cells, or they become effector cells which can take effect immediately. Rapid and most stronger response is produced later because of these memory cells. So here you have to remember about the effector B cells and the effector T cells, the effector form of the B cells which are the plasma cells, effector form of the T cells which are the helper T cells and the cytotoxic T cells. Now the effector form of the T cells or the T lymphocytes are the cytotoxic T cells and the helper T cells. Cytotoxic T cells, they produce toxic proteins which kills the infected cells. They just kill the infected cells. Okay. This helper T cells, they can produce signals which activate these cytotoxic T cells, which in turn produces the toxic protein to kill the infected cells. So either they can directly kill the cells, or some of the helper T cells can produce signals which activate these cytotoxic cells to kill the infected cells. So that is one part by which they act, and the second part is like this, where the helper T cells, okay, they produce signals. They can also produce signals to activate the B lymphocytes. Okay, so these helper T cells not only can activate the cytotoxic T cells, but they can also activate the B lymphocytes. Okay, and these B lymphocytes, they can initiate the production, antibody production. Okay, these can produce these antibodies which are, sent into the blood and lymph, and they can mark the antigen for inactivation and destruction. Okay, they can also, these B lymphocytes can also activate what are called the plasma cells. Plasma cells are the effector form of the B lymphocytes that we seen in the other slide. The effector form of the B lymphocytes are the plasma cells. So these plasma cells, they undergo a lot of this proliferation to form a clone of cells, and then they secrete these antibodies into the blood which can take action against the pathogen. So remember that the B lymphocytes, they don't kill the cells, but they mark the cells for inactivation and destruction. Okay. Now the elimination of the invader. So the effector form of T cells are the cytotoxic T cells and the helper T cells. That's what we seen in that that chart. Okay. And the cytotoxic T cells, they use a toxic protein to kill the infected cells, and signals from helper T cells, they activate the cytotoxic T cells to kill the infected. So the explanation for that chart is here. So signals from helper T cells, they can also activate the B lymphocytes to initiate anti-production, all that. and effector form of B lymphocytes are plasma cells. So a single activated B lymphocyte can form thousands of plasma cells. And plasma cells, they secrete the soluble form of B lymphocyte antibodies, antigen receptor, in large quantities, which are released into the blood and lymph. So this is the explanation for the previous chart. So the circulating antibodies can neutralize and inactivate the toxins and pathogens in body fluids. Okay, so they don't kill. So the B lymphocytes, they don't kill the cell. They don't kill the pathogen. The B lymphocytes, they don't kill the cells. They don't kill the cells, but they can neutralize and inactivate the toxins and pathogens in the body fluids. Okay. So action of a cytotoxic T cell or infected host cell, you don't have to know about these granzymes and all that, but you can see this cytotoxic T cell binding to the infected cell and producing some enzymes, enzymes which form pores in this, and they produce some toxic substances which kill the infected cell. Okay. Now, activation of B cells in the humoral immune response. So here we see in the antigen presenting cell, helper T cell binding to that, and the activated helper T cell. Once it is binds to the B cell, it gets activated, the cytokines are produced, and then the B cell can so the T cell can also activate the B cell. That's what we seen in that particular chart. You can refer the chart and see the T cell, when it is activated by binding to the T cell binds to the pathogen, the antigen fragment, then it becomes activated, and the B cell can produce some plasma cells which secrete antibodies, and some of them become memory cells. So that is the same thing which is happening in the immune response. So these antibodies only, this is the antigen receptors. No? So the antigen receptors are the antibodies which can bind to the antigen and inactivate it. Okay. Now, immunological memory, they provide immunological memory. So the following differentiation into effector T cells, cytotoxic T cells and helper T cells, the T lymphocyte in the clone remain as memory cells. Now the cytotoxic T cells and helper T cells, they are the effector cells. Then I told you some other cells also form in the clone, and they remain as memory cells, which are long-lived, and if they confront the same pathogen or antigen later in life, they can become, or they give rise to the effector T cells later in life. Okay, the same antigen is encountered later in life. So similarly, the remaining B lymphocytes in the clones are memory B cells. So same thing for the B cells also. They are long-lived and give rise to plasma cells. The same antigen is encountered. Okay. So these memory T cells and memory B cells, they cause a stronger and rapid response at subsequent encounter of the same antigen. So this is the immunological memory. So this immunological memory is called secondary immune response. Okay. So this immunological memory is called a secondary immune response. Here there is a chart about this immunological memory. So you have the T cells here, okay, and the active form of the T cells, or the helper T cells, the cytotoxic T cells, which take effect immediately, and memory cells also are formed, which are long-lived, and they can give rise to effector cells if the same antigen is confronted. They meet the same antigen later in their life. B cells, they also produce the effector cells, which are plasma cells, and memory cells also are produced if the same antigen is confronted later in their life. Now, this is a summary of this humoral immune response and the cell-mediated immune response. Now, this is the first exposure. Okay, first exposure to the antigen. The antigen is engulfed by an antigen-presenting cell. Okay. Then what happens? This antigen-presenting cell, it activate once the antigen binds this antigen presenting cell, it displays that antigen fragment, and then this T cell can bind to it, helper T cell can bind to it. Okay, so it can form the cytotoxic T cells. Okay, or it can even give rise to, or it can activate the B cells. The helper T cell can activate cytotoxic T cells, or it can activate the B cells. Okay. Directly also the antigen, when it binds, the B cell is activated. Or it can, or this way also it can, the T helper T cell also can activate the B cell. Or it can activate the cytotoxic T cell. Okay. So now what happens here, the B cell, it produces proliferate and form a clone of plasma cells which are the effector cells, and they secrete the antibodies into the blood and lymph, and which can destroy or inactivate the pathogen. The cytotoxic T cells, this activated cytotoxic T cells, what they do is they can destroy the cells. They kill the cells which are infected by the pathogen. Okay, and what happens? Some of these cells in the clone, they become memory cytotoxic T cells, and some of these become memory B cells. Okay, so memory helper T cells also can be formed. Okay, so second exposure to the same antigen, what will happen? These memory T cells activated, which activate the memory cytotoxic T cells and memory B cells. Okay, and what will happen? They can produce the effector plasma cells or the effector cytotoxic T cells, which can produce a stronger and rapid response to the pathogen. Okay.